So this is the last video of the series about DNA for Chapter 3. I'm going to be talking about DNA replication today, and this corresponds mostly with 3.4 in your textbook, but I'm going to make some references to some other sections to make some connections for you. But before we actually get started, we need to talk about why DNA replication is so important. What you should know is that DNA replication is the process of making copies of DNA. And really what we're talking about is, when we're talking about DNA, we're talking about deoxyribonucleic acid. We're talking about genetic information. And we talk, really, when we're talking about genetic information, we're talking about genes. And a gene is a section of DNA that codes for a specific protein. Sometimes we think about them as traits, and we'll come back to that, but come back to this idea later. But that's really the importance, is if we have the same DNA in every single cell, then we have the same genetic information, and therefore each cell could express the protein that it needs to in order to survive or do its job. So if we think about this in the context of ourselves, remember that when we started our development, we started from one single cell, and it divided. Now this is divided through mitosis to other cells, and they kept dividing, and those cells kept dividing until we got to the trillions that we have today. The trillions of cells that we have today. Now they all have the exact same, for the most part, I'll say exact, quote unquote, exact same DNA, aside from any mutations, that was in that first single cell. And that DNA had to be copied before those cells could go through mitosis, and when it gets copied, that genetic information can be carried to every single cell. Now you might be asking, why do my cells look different? Why do my eye cells look different than my heart cells? Even though they have the same genetic information, they're reading different genes within that same sample. So my analogy for that is kind of like that every single cell is carrying around the entire set of Encyclopedia Britannicas in hard copy, but only reading one book out of the whole volume, out of the whole series. So they're carrying around all the information, but only reading small sections of that, depending on what kind of cell we're talking about. So this is really the key idea of why we need DNA to be copied, is so that each cell can make the protein it needs. And remember that proteins are made through this process of transcription and translation. And so remember that DNA's message gets converted to RNA's message, which gets converted into proteins by putting amino acids together. And this, I guess I'll do this in another color. This is what we call the central dogma of biology. So this is why we studied genetic information so much. It's really about making proteins. And this is really the key idea here behind this molecular biology. But let's go and get started and talk about DNA replication itself and how it happens. So we got to talk about a couple of enzymes, in particular helicase and DNA polymerase, which interestingly are, just like we've seen helicase before in transcription, and we've seen RNA polymerase in transcription, which are proteins which are involved in making other proteins. We're going to talk about helicase again and DNA polymerase this time. And then we're going to talk about complementary base pairing one more time and semiconservative replication. So, what I have over here is a section of DNA. It's a strand of DNA. It's missing some stuff. So let's go ahead and do the proper base pairing. And remember that base pairing rule is that A bonds has 2 bonds to T. I don't know, maybe just writing it again is helpful. And C actually has 3 triple bonds to G. So these are the proper base pairing rules. Just so you know, so we can give credit to where credit is due. These are Chargaff's base pairing rules. We're talking about DNA, so we're talking about thymine here. So G bonds to C, A to T, T to A, T, G, T. And if you notice, I've just repeated the same sequence. It's just a random sequence. So go ahead and copy that down again. And one more, and again. I think there's one more actually. Okay, so I've got my complementary base pairs. There are hydrogen bonds between these. And remember that these hydrogen bonds are weak, so they're going to break much faster than the covalent bonds. But that reminds me that we should point out that this line right here is, well, we draw that to represent the sugar and phosphate backbone. So remember that every nucleotide, and here we would say is one nucleotide. Well, these are the monomers to make up the polymer of DNA. One nucleotide is a sugar, deoxyribose sugar, a phosphate, and a nucleotide. A nitrogenous base. So T is my nitrogenous base in this example of a nucleotide, but the sugar and phosphate are going to be the same for every single nucleotide in DNA. It's always going to have deoxyribose and always going to have a sugar. So here we can say this is the sugar-phosphate backbone. We don't have to draw it every time because really the key difference is going to be the nitrogenous bases. So that's what we highlight. So the lines represent the sugar-phosphate backbone. But look back at the structure of DNA and the podcast. There are the 5 carbon sugars and the 3 carbon sugars are used to indicate, here what we talk about is 5' and 3' end. So this end is a 5' end, which actually makes this end the 3' end. This end is the 3' end. This is the 5' end. Because you remember the DNA is anti-parallel. One side is flipped over and goes the other direction. So I want you to look at back at our drawing of from DNA structure of your drawing of DNA and see if you can figure out which end is the 5' end and which end is the 3' end. But know that they're opposite. The 5' end is the one that has at the very end of it the 5 carbon sugar with a phosphate, a lone phosphate that's not attached to another nucleotide, and this is one that has a free 3 carbon sugar, and vice versa on the other side. So we've got this DNA structure, and what actually needs to happen, I'm going to remove a few things here. Okay, so, sorry, I'm going to move a couple of things. So what I'm going to point out, I removed some of the layers, some of the additional notations I put in. Our first enzyme that we need to know about is called helicase, and I'm going to represent it here in this purple. This is helicase, and remember this is an enzyme, and it works on the helix. Remember this is a double helix. Sorry, let's do it in a color that's color-coded. So this is the double helix. Think of it as it has spun around itself. but it's laying flat to show this process. Helicase comes in, it's moving in the right to left direction, which means that this DNA has been unzipped. So these single strands are now separated. They're single strands of DNA, which is a problem. Here, when it's double-stranded, it has a check against itself to carry the same message. T always has to bond to A always has to bond to T. But when it's single-stranded, it's a little more vulnerable. That means that if one of these switched for whatever reason, beyond my understanding, there's not something to check against itself to make sure it copied it correctly. So, in other words, it's as if you have copied something down from the textbook, but then you no longer have the textbook and now are unsure of whether you copied it correctly. So anyways, that's one of the leading things of mutations. But anyways, helicase unzips, and then what ends up happening is we add another enzyme, and I'm going to use a bluish color to represent the DNA polymerase. So DNA polymerase is following behind the helicase, and what it's doing is adding nucleotides to complement the ones on the single strand. So we know C bonds to G, A to T, T to A to T, T to A, and so on, all the way down the line. So that will continue all the way down here, and as the helicase continues to move, this DNA polymerase will follow. Remember, DNA polymerase is another enzyme with the ASC ending, and it makes new DNA polymers by putting DNA nucleotides together. Now, these DNA nucleotides are floating around in the nucleus, in the cytoplasm. If we're talking about it's a nucleus if it's a eukaryotic cell, but it's floating around inside the cell, and they're coming from the food that we eat. We've digested the DNA from the cells that we've eaten, broken them up to their monomers, and use them for our own growth. So if we weren't eating, we wouldn't be able to make new copies of DNA, and therefore not be able to make new copies of cells. Okay, but they're floating around in the cytoplasm, and they get pulled in by DNA polymerase and attached and go all the way through. Now, what's interesting is that DNA is formed in the 5' to 3' direction. So we said that this top part here is the 5' end, this is the 3' end. So that makes this the 3' end, this the, whoops, do this in green, I apologize. This the 5' end, and as it moves down, it's adding on the 3' end. So the DNA sequence gets added continuously. Now, what's interesting is on the other strand, we've got a 3' end here and a 5' end here, which means that this is the 3' end, sorry, that means that this down here would be the 3' end, and this would be the 5' end. Now, so the DNA is actually going to be created in the opposite direction. So it's going to be going this way. So our other molecule of DNA polymerase will be traveling this way, adding in new nucleotides, base pairing, following the same rules of Chargaff, A's to T's, C's to G's as it moves down the line. Okay? Now, one key difference between this process of helicase and DNA polymerase, one key difference between that and transcription, which is making RNA, is that this is happening on both strands, whereas mRNA is only being made from one of the two strands. And this happens in several places at things that we call places that we call replication forks. And it can happen several places. So I'm asking you to remind me, because I'm likely to forget, to show you a model of this in class to help you understand that this can happen in lots and lots of places. Okay? So that being said, I think that that's the process of DNA replication. So you also need to explain the significance of complementary base pairing and the conservation of the base sequence of DNA. Well, I would ask you to go ahead and write out all of the base pairs, practice and base pairing, and what you'll see is that this sequence of DNA that we're looking at right here is an exact copy. The top section is an exact copy of the bottom section. So really what's important to recognize is that this base pairing allows for exact replication of DNA because it's providing a template across which it could add new bases to carry the same message. So if we've got, the same DNA, then we've got the same genes in after every single DNA replication. I'm not saying that we all start with the same DNA sequence. We most certainly don't unless we're identical twins. But these same genes get copied into every single cell so that later transcription and translation to make proteins can still occur in the way that the DNA, I guess, quote unquote, intended to when you inherited it. So that's really the significance of DNA, of complementary base pairing, is to make the exact same copy of DNA and serve as a template. So we need to talk about DNA being semi-conservative. And we kind of danced around this idea, but let's just define it. So semi-conservative. All right, conservative, I mean, aside from political connotations, conservative, to conserve means to save, right? So if we think back to our model of DNA replication, think about the new strand of DNA. So let's look at just this section right here. How much of this sequence was saved from the original sequence that we see here? Well, you should recognize that half of that DNA sequence is this top line. This is the old strand, and this one down here is going to be the new strand. So some of it gets saved, that's the conserved part, and semi means half. So the idea is that with every new strand of DNA, half of it is actually part of the old strand from which it was copied. Now, of course, when you talk about the next cycle of DNA replication, we reevaluate, but basically it's every time DNA replication happens, half of it's from an old strand and half of it is newly synthesized or newly made. And that's what semi-conservative means. And again, it allows for identical, as much as possible, identical replication of the DNA sequence, which means we've got the same genes and transcription and translation occur accurately. Now, I want you to, before I end this, I want you to think about what would happen if one of these nucleotides changed to a different nucleotide with a different nitrogenous base, and what the implications of that are for transcription and translation, and making proteins and amino acid sequences, and how that might impact the actual shape of the protein that's made, either positively or negatively. And if you're totally lost about what I'm talking about, please ask me, because that's the whole point of this section of the course. Okay, and I'll leave you here.